Dear ladies and gentlemen, today I want to present you my speech about application bioinformatics in autoimmunity disorders for identifying druggable pathways and promising targets. Mankind had been started to use biomarkers long before it was named, with the emergency of desire to know if the person is alive or dead, ill or healthy. It was common for bleeding, fever, and turned blue skin to be the character of diseases. However, all this is a typical sign to a large number of pathologies, and so it was necessary to observe and systemize the characteristics of individual diseases. The novel biomarkers are considered to be synonymous with molecular markers. Targeted therapy is based on the binding of target molecule or cell, which blocks or modifies the action of the target. It leads to the restoration of the physiological state. A target is a range of biological entities which may include oncocells, proteins, genes, and relationship between microRNA and mature RNA, and so on, just as long as they have something to do with certain diseases. The basis of this treatment is drug discovery, the process by which drugs are designed or discovered. Roughly speaking, the entire drug discovery process includes the following 5 phases. Target identification, target validation is crucial for our purpose. Such process from the original idea to creating finished product takes from 12 to 15 years and costs about $1 billion. Past records have indicated that the high failure rate, only 8% of success of drug development, can be largely attributed to improper target selection. As such, target discovery process is most crucial step in a modern drug discovery campaign. Although the pioneers of computational biology in 1960s did not use the term bioinformatics while reporting about their works, they had a clear vision of how computer technology, mathematics, and molecular biology could be successfully combined to answer fundamental questions in the life sciences. One of the first steps of introduction to bioinformatics was written by Margaret Dayhoff program, which allows to work with the overlapping peptide fragments from the digestion of a protein in puzzle-like manner to determine the amino acid sequence of protein molecule. After that, she constructed the atlas of protein sequence and structure, served as the first database for molecular biology. It became an indispensable, inspiring source for early computational research. Today, bioinformatics are formed as an independent interdisciplinary science, and it is no longer just a tool for data processing and interpretation of the results of omics analysis. Its personal goal is to work with big data, that is really hard task for human alone, and to search for a new, non-trivial information. The latter goal is defined as data mining, which allows to form a new or modified present approaches to disease diagnosis and therapy. Data mining is the process of analyzing data from different perspectives and summarizing it into useful information. Data mining software allows users to analyze data from many different dimensions or angles, categorize it, and summarize the relationship identified. Data mining of available biomedical data has led to significant achievements in target identification. In this context, data mining refers to the use of a bioinformatics approach not only for the identification, but also for selecting and prioritizing potential targets. Initially, the method used to analyze data were mostly statistical, but the application of machine learning tools to biomedical problems gives a chance to make a significant breakthrough, especially in classification tasks that in the particular fields are diagnostic disease or control group and types of illness, and drug response monitoring via short time series of gene expression. Machine learning approach allows instead writing a program by hand for each specific task to collect a set of samples training data which specify the correct output for a given input. A machine learning algorithm takes these examples and produces a program which may work with other sets perfectly. One of the most perspective data mining approaches, which provides a wide range of tools, is using R language. It is a programming language and environment which includes a plenty of statistical and graphical techniques. There is also a bioconductor, a package built on R language which provides tools for the analysis and comprehension of high throughput genomics and proteomics data. It has two released each year and an active user community. But I must tell you that a lot of R packages don't have a GUI, graphical user interface, and it is not so easy to apply such packages for biologists. But a lot of packages have this interface. Macroarray R as leverages cutting-edge bioconductor packages for researchers with no knowledge in R language. It also enables a more reliable and accurate macroarray data analysis and expedites downstream functional analysis of macroarray results. It includes GUI for plenty of macroarray analysis software. For Illumina data, for example, the software accepts pre-processing data output from Genome Studio and supports fully customizable pre-processing with Illumina package for non-pre-processing data. In one study, computational macroarray analysis of peripheral blood mononuclear cells samples from children with T1D, including groups new 1-month diseases, 4-month diseases, and healthy control group was used. And a Lima package in R language was applied for this research. It is enough to identify the differently expressed genes, but if we want to explore changes in biological process, we should continue such research. So the online software database for notation, visualization, and integrated discovery may be used to perform pathway analysis for the differently expressed genes. It is also allowed to apply iPath Guide, which brings the most advanced pathway analysis platform to web. In study we review earlier, p-value less than 0.05 was defined as a cutoff to identify the statistical significance of the biological pathway that differently expressed genes are involved in. Several properties of PPI, protein-protein interaction networks, such as allosteric sites and hotspots, have been incorporated into drug design strategies. And PPI is suggested to be helpful in discovering putative therapeutical targets for the development of new treatment. Construction of PPI network may be performed with the help of String, Cytoscape, PAGIC, or other PPI databases. Cytoscape is an open source software platform for visualizing molecular interaction network and biological pathway and integrating this network with annotations, gene expression profiles, and other state data. Additional features are available as plugins. Plugins are available for network and molecular profiling analysis, new layout, additional file format support, scripting, and connection with databases. In the study we reviewed, don't forget it was about children with T1D. A resulting network was built with the help of String, and there is 35 nodes and 61 edges in this network. Then the node degree of the network was analyzed, and the results show that 5 hub proteins was identified. They were first proplated basic protein, interleukin 8, and so on. They may be applied for further research as potential targets for therapy. Bioinformatics study indicated that more than 60% of human genes may be regulated by microRNAs. So it opened to the big gate to change genes expression with the help of microRNAs. Though the tens of different methods had been developed for computational target prediction. I want to tell you about several of the best. Target scans, Picture, and Diana MicroT. Target scans is an algorithm developed to identify the targets of vertebrates' microRNAs. This program uses computational sequence analysis to predict microRNA targets conserved across multiple genomes, such as human, mouse, rat, duck, chicken, and pufferfish. Contrary to target scans that require seed match and at exactly corresponding position in a cross-species uterine, Picture require binding sites that are co-regulated by multiple microRNAs across species. Picture checks the alignment of friend ureters for those displaying seed matches to microRNAs, filters the retained alignment based on their thermodynamic stability, and computes maximum likelihood score for each predicted target. Autoimmune myocarditis is an inflammatory process of the myocardium which result in injury to the cardiac muscle cells, and the manifestation range from subclinical to sudden death. It may be induced by infection, toxic, or immunology agents. One of the most common infection agents causing myocarditis is Coxsackie virus B3. One experiment in cell culture and then in mice to study the dynamic of 50 microRNAs was carried out. In that study, it was found that the expression of microRNA 151 and microRNA 148A was significantly upregulated. Since microRNAs regulate biological processes but by repressing target genes expression, it was predicted that target genes of microRNA, these both microRNA, by using online bioinformatic tools TargetScan and Miranda. It was relay, an important component of nuclear factor Kb signaling that was confirmed in further experiment. So we may conclude these two microRNA as potential targets for modulate immune response to Coxsackie virus B3 and to protect the body from overdriving immune action and therefore to avoid development of myocarditis. Traditional cardiac risk factors do not explain the high incidence of SLA, although the risk of cardiovascular diseases associated with SLA in 4 to 8 times higher than in the normal population. In a 2014 research, it was investigated whether traditional risk factor and some of the biomarkers candidates could be combined to create an overall risk profile using self prediction modeling software. This software creates multiple classification trees for prediction, identifies those independent variables that best segregate as important predictors, and identifies the most predictive cutoff point for each independent variable. Autodemographic variables, traditional cardiac risk factor, markers of SLA disease, activity and damage, and biomarkers were entered into the software. The variables cutoff points that were identified as most predictive for plaque were then involved with univariate and multivariate regression analysis. The following variables were significantly associated with carotid plaque in both the SLA group and the control group. Using Salford predictive modeling software, the most significant independent predictors and cutoff points was as follow. So it was found that the predicts panel of 4 inflammatory biomarkers and 2 traditional cardiac risk factors, age and diabetes, as compared with individual biomarkers or risk factors, had overall better predictive capacity for the presence, progression, and ischemicity of carotid artery plaque in SLA patients who were followed up for 2 years. The predicts profile also demonstrated a better predictive capacity than a panel of traditional cardiac risk factor. Thus, predicts is a good instrument for identifying SLA patients at increased risk of developing it. However, no single variable demonstrated an ideal combination of good negative predictive values or positive predictive values, sensitivity and specificity, so that there is basis to suggest that such panel may be a step to identify closer relations these two diseases and to find druggable pathway in order to prevent significant cardiovascular disorders. Passing through these approaches, I hope you judge rightly about bioinformatics indispensability. And if you still have not used its approach a lot in your lab, I advise you will start immediately. Now you may understand why. Thank you for your attention.